Heredity and Evolution, Part 1. We have seen that reproductive processes give rise to new individuals that are similar but subtly different. We have discussed how some amount of variation is produced even during asexual reproduction. And the number of successful variations are maximized by the process of sexual reproduction. If we observe a field of sugarcane, we find very little variations among the individual plants. But in a number of animals, including human beings, which reproduce sexually, distinct variations are visible among different individuals. In this chapter, we shall be studying the mechanism by which variations are created and inherited. The long-term consequences of the accumulation of variations are also an interesting point to be considered. We shall be studying this under evolution. Accumulation of variation during reproduction. Inheritance from the previous generation provides both a common basic body design and subtle changes in it for the next generation. Now, think about what would happen when this new generation, in its turn, reproduces. The second generation will have differences that they inherit from the first generation, as well as newly created differences. As shown here, this figure would represent the situation if a single individual reproduces, as happens in asexual reproduction. If one bacterium divides, then the resultant 2 bacteria divide again. 4 individual bacteria generated would be very similar. There would be only very minor differences between them, generated due to small inaccuracies in DNA copying. However, if sexual reproduction is involved, even greater diversity will be generated. As we will see when we discuss the rules of inheritance. Heredity and Evolution, Part 2. Environment in which they find themselves. Obviously not. Different kinds of advantages. Bacteria that can withstand heat will survive better in a heat wave, as we have discussed earlier. By environmental factors form the basis for evolutionary processes, as we will discuss in later sections. Let us now understand the concept of heredity. Heredity. The most obvious outcome of the reproductive process still remains the generation of individuals of similar design. The rules of heredity determine the process by which traits and characteristics are reliably inherited. At these rules: inherited traits. What exactly do we mean by similarities and differences? We know that a child bears all the basic features of a human being. However, it does not look exactly like its parents, and human populations show a lot of variation. Rules for the inheritance of traits. Mendel's contributions. Part 1. The rules for inheritance of such traits in human beings are related to the fact that both the father and the mother contribute practically equal amounts of genetic material to the child. This means that each trait can be influenced by both paternal and maternal DNA. Thus, for each trait, there will be two versions in each child. What will then the trait seen in the child be? Mendel worked out the main rules of such inheritance, and it is interesting to look at some of his experiments from more than a century ago. Mendel used a number of contrasting visible characters of garden peas, round or wrinkled seeds, tall or short plants, white or violet flowers, and so on. He took pea plants with different characteristics, a tall plant and a short plant, produced progeny from, and calculated the percentages of short or tall progeny. In the first place, there were no halfway characteristics in this first generation or F1 progeny. No medium height plants. All plants were tall. This means that only one of the parental traits was seen, not some mixture of the two. So the next question was, were the tall plants in the F1 generation exactly the same as the tall plants of the parent generation? Mendelian experiments test this by getting both the parental plants and these F1 tall plants to reproduce by self-pollination. The progeny of the parental plants are, of course, all tall. Rules for the inheritance of traits. Mendel's contributions. Part 2. However, the second generation, or F2, progeny of the F1 tall plants are not all tall. Instead, 1/4 of them are short. This indicates that both the tallness and shortness traits were inherited in the F1 plants, but only the tallness trait was expressed. Thus, 2 copies of the trait are inherited in each sexually reproducing organism. These two may be identical or may be different depending on the parentage. A pattern of inheritance can be worked out with this assumption, which is as shown. In this explanation, both TT and TT are tall plants, while only TT is a short plant. In other words, a single copy of T is enough to make the plant tall, while both copies have to be T for the plant to be short. Traits like capital T are called dominant traits, while those that behave like small t are called recessive traits. However, the question arises, what happens when the pea plants showing 2 different characteristics rather than just one are bred with each other? What do the progeny of a tall plant with round seeds and a short plant with wrinkled seeds look like? They are all tall and have round seeds. Tallness and round seeds are thus dominant traits. But what happens when these F1 progeny are used to generate F2 progeny by self-pollination. A Mendelian experiment will find that some F2 progeny are tall plants with round seeds and some were short plants with wrinkled seeds. However, there would also be some F2 progeny that showed new mixtures. Some of them would be tall but have wrinkled seeds, while the others would be short but have round seeds. Thus, the tall or short trait and the round seed or the wrinkled seed trait were independently inherited. Rules for the inheritance of traits. Mendel's contributions. Part 3. How do these traits get expressed? How does the mechanism of heredity work? Cellular DNA is the information source for making proteins in the cell. A section of DNA that provides information for one protein is called the gene for that protein. How do proteins control the characteristics that we are discussing here? Let us take the example of tallness as a characteristic. We know that plants have hormones that can trigger growth. Plant height can thus depend on the amount of a particular plant hormone. The amount of the plant hormone made will depend on the efficiency of the process for making it. Consider now an enzyme that is important for this process. If this enzyme works efficiently, a lot of hormone will be made, and the plant will be tall. If the gene for that enzyme has an alteration that makes the enzyme less efficient, the amount of hormone will be less and the plant will be short. Thus, genes control characteristics or traits. If the interpretations of Mendelian experiments we have been discussing here are correct, then both the parents must be contributing equally to the DNA of the progeny during sexual reproduction. We have discussed this issue in the previous chapter. If both the parents can help determine the trait in the progeny, both the parents must be contributing a copy of the same gene. This means that each pea plant must have two sets of all genes, one inherited from each parent. For this mechanism to work, each germ cell must have only one gene set. However, the question arises, how do germ cells make a single set of genes from the normal two copies that all other cells in the body have? If progeny plants inherited a single whole gene set from each parent, then the previous experiment cannot work. This is because the two characteristics R and Y would be then linked to each other and cannot be independently inherited. Rules for the inheritance of traits. Mendel's contributions. Part 4. This is explained by the fact that each gene set is present not as a single long thread of DNA, but as separate, independent pieces, each called a chromosome. Thus, each cell will have two copies of each chromosome, one each from the male and female parents. Every germ cell will take one chromosome from each pair, and these may be of either maternal or paternal origin. When two germ cells combine, they will restore the normal number of chromosomes in the progeny. Ensuring the stability of the DNA of the species. Such a mechanism of inheritance explains the results of the Mendel experiments and is used by all sexually reproducing organisms. But importantly, asexually reproducing organisms also follow similar rules of inheritance.